Thank you very much. I appreciate it. And thank you, Anna and Hunter's Hope, for having me here to talk about a little bit about what I do at Travere. It's kind of a unique position to have somebody focused solely on diagnostics. And particularly, I'm going to be speaking about how Travere supports shortening the diagnostic odyssey in rare disease. Can we have the next slide, please? I'm just going to tell you a little bit about us. Travere is based in San Diego. We're a biopharmaceutical company that specializes in identifying, developing, and delivering life-changing therapies for people living with rare disease. We have 3 commercial products, Chenodal and Cholbam. We look at for patients who have bile acid synthesis disorders. Specifically, we look for CTX, cerebrotendinous xanthomatosis, and Zellweger spectrum disorder. We also have a drug Thiola, which is for cystinuria. And we have a couple of pipeline products for rare nephrology conditions, and pegtibatinase is coming up for classical homocystinuria. We're at about 350, moving towards 400 employees, and we have offices in Ireland and Switzerland. Next slide, please. Our mission, as I mentioned, it's to identify, develop, and deliver life-changing therapies to people living with rare disease. We have a vision to be a leader in the rare space and give hope to patients in the global rare disease community. We focus on our values to be patient-inspired, be courageous, and promote the community that we know that we're all better and we're stronger together. Next slide. And at the center of everything we do, and we don't just say it, we do it every day, is the patients. The patients are our why. The patients are what we do every day. And that's where my job comes in, is to try to do what I can to shorten that diagnostic odyssey for all the rare disease patients. Next slide. So I talk about my job as a 360-degree diagnostics approach. And what does that mean? That means we look under every rock. We use biochemical testing, we use genetic testing, we support research projects, we're involved in newborn screening. And so my job as not only monitoring all of the operations of the testing programs is to also look at strategies for the future of ways that we can find these patients better and faster. Next slide. So for example, for CTX, we have 6 different initiatives, biochemical and genetic testing programs. We support the New York Screen Plus project. I'll talk about these in future slides, but I always like to look at it that we come at diagnosing these patients in a variety of different ways, trying to find them because they may not be in a cholestasis panel, but they may be found in a bilateral cataract panel. Next slide. So for Zellweger spectrum disorder, it's the same idea. We have different programs, and we're always looking to see what might be the next opportunity to find these patients. Next slide. So we have a number of what we call no cost to patient sponsored testing programs. We don't call them free testing programs because nothing is free, but the goal of what we do is to try to increase access to this testing and try to provide earlier diagnosis to patients. It's also helpful for providers as well. They don't have to go through insurance. They don't have to worry about reimbursement or anything. They just have the kit, they fill out the requisition form, and they send it to the laboratory and they get the report. Next slide. We have 2 biochemical testing programs, some of our oldest testing programs before I started at Travere at Cincinnati Children's Medical Center. Dr. Ken Setchell and Dr. Jim Hybe were actually the developers of Cholbam, which is one of our commercial products. And Dr. Setchell is the laboratory director who developed and validated a urinary bile acid profile analyzed by what they call fast atom bombardment mass spectrometry. And that actually provides a diagnosis to patients who have cholestasis or unexplained liver disease. And in that test, we're looking for bile acid synthesis disorders and CTX. The issue with that testing program is that it's semi-quantitative, it's a little bit subjective, and it's only performed at Cincinnati. So Travere has been working with Dr. Setchel over the last couple of years for him to develop and validate a quantitative assay that is almost complete so that other people in his lab could run the testing program and other labs could do it as well. But it also will allow for dose adjustment for patients, which we always feel is a kind of a gold standard because patients are different and what kind of dose is working the best for them in their condition is always important. They also have a Zellweger spectrum atypical bile acid test, which is a serum-based test. And because those are toxic to the liver, it's also not only used for diagnosis, but it's used for treatment monitoring because we want to make sure that those bile acids are at a low level for those patients. Next slide. Then we have testing programs with Dr. Andrea DeBarber, who you heard from yesterday at Oregon Health and Science University in her sterile analysis lab. We worked with her to develop these biomarkers in plasma and urine for biochemical testing for CTX that we used in one of our clinical trials. And then we moved on and we gave her some extra funding to actually clinically validate them in her clinical laboratory. So that testing program is used for biochemical confirmation for CTX, but it's also used in therapeutic monitoring purposes at different points after therapy is initiated. And we feel like this is an incredibly important piece, especially when we start talking about newborn screening for CTX, which she was talking about yesterday, because when we talk about babies, infants, children, as they continue to grow, it's going to be really important to make sure that their dose is adjusted appropriately. So this is it's a comprehensive biochemical testing program. We also have a diagnostic program that's a dry blood spot testing program that she developed and clinically validated for 7-alpha-12-alpha-C4, which is elevated in patients with CTX. And then we have a genetic confirmation at Molecular Vision Lab, which is also in Oregon. Next slide. So all of our genetic testing programs are now at Prevention Genetics. The cholestasis panel is the oldest program that Travere has supported. We started at Emory Genetics Lab and moved it to Prevention last year. And at this point, we have reported over 10,000 samples, which is pretty amazing. Greater than 50% of those samples are in infants less than 6 months old. A lot of these babies are cholestatic. They're in the NICU and the PICU, and it's kind of become standard of care for pediatric hepatologists and gastroenterologists to use this program. One of them told me that sometimes it at least gives him something to tell the patients that he is actually doing something. Sometimes the liver resolves and the patient goes home before genetics is even done, but at least they're creating the process and we've made it really easy for them to use. It's got 66 genes on it. Now it's 77, which includes CTX and Zellweger's. But we've actually find way more other diagnoses besides ours. So we feel good about providing the service to this community. The next program is called Behind the Blur, which is an idiopathic bilateral cataracts program based upon a prevalence study that we did on cataracts, which, as I mentioned, the 360-degree approach, we do the research and say this is a good community to target for genetic testing. It's got 66 genes, including CTX. We're looking at patients between 18 months and 35 years. And since Q3 of 2020, we have found a 19% positive rate. We're getting ready to, we've had an abstract submitted and we're going to publish that. The good news is that most of the conditions are not life-threatening. They're crystalline genes that can just, have the cataracts taken care of. But we're pointing to pediatric ophthalmologists, they're a little bit leery about using genetic testing. So we want to make sure that they're comfortable having this program in hand. And we just found out actually yesterday, we just found our first CTX patient through this program. It's a 9-year-old kid. So I think that it's worth all of what we're doing because early diagnosis is certainly really key in CTX. We have a PVD-ZSD national program, which is just for diagnosed or clear clinical suspicion of Zellweger's. That is just the PEX genes. And then the last program is what we call the XALD newborn screening reflex program, which we saw a gap once ALD came on the RUSP to be screened in the States, that there are other conditions, including Zellweger's and Aicardi-Gouttières syndrome, and some other conditions that actually present with the elevated very long chain fatty acids. And if they're just going to confirm for the ABCD1, we may miss some of these patients. So we're offering a reanalysis of ABCD1 and the other genes as well, in order to catch those babies if they're not caught in the States right at the get-go. Next slide. Travere also supports a number of research projects. I mentioned, in order to go into these sponsored testing programs, we need to have the data to support that. So we support some of these extra projects. Next slide. One of them is a collaboration between OHSU with Dr. DeBarber and Western Galilee Medical Center in Israel with Dr. Philippe Zakai. They've been screening dry blood spots on babies in Israel in a high-risk Druze population, doing biochemical testing and genetics since about 2014, 2015. And a lot of the purpose is so that we can use this high disease prevalence population in newborn screening to allow for a faster evaluation of the dry blood spot method that Dr. DeBarber has developed to look at the sensitivity and the specificity. And this project is going to be another piece of the support for CTX on the RUSP nomination in the United States. And that project is almost complete. Next slide, please. We're also supporting an ataxia study. So ataxia is frequently seen in 62% of CTX patients. So again, kind of like the cataract program, we're looking at CTX in ataxia. Would this be a good diagnostic strategy? We need to just try it. So the University of Alabama is the specialty clinic is screening ataxia patients using the dried blood spot program that we have at OHSU with Dr. DeBarber. And the goal there is 100 patients. So again, that may turn into a diagnostic strategy down the road where we actually screen ataxia patients for CTX. Next slide. This is a project that I lead, and I call it the Genetic Testing Landscape Project. The goal is to get all of our genes of interest on appropriate genetic panels in major genetic testing labs in the United States. We have clinical account managers who go out and talk to providers about our company and our conditions and our treatments. And what they found was sometimes a doctor would say, oh, well, I just sent a test to Invitae for an ataxia panel, and they look at the website and they find out that CYP27A1, which is for CTX, is not on the panel. So they asked if there could be a way for me to work to try to increase our numbers on genetic panels. So I hired an external company to assess US labs, 24 labs. They gave me the spreadsheet, and then phase 2 and phase 3 is the follow-up, which has been really, really challenging because a lot of labs have different methods and timelines for addition of genes. But I just did the data for the phase 2 results, and we actually have seen a 23.3% increase in CTX and Zellweger spectrum disorder genes on panels across the United States. So no matter where the patient comes into the healthcare system, no matter where a provider sends the genetic testing to, we know that these patients will get picked up if they're actually there. Next slide, please. Newborn screening projects. Next slide. We've talked about this before. Melissa Wasserstein and the New York Screen Plus, 175 babies looking at conditions outside of the RUSP, including CTX. And this is critical for the RUSP resubmission here in the United States. So Travere is one of the sponsors of that project. Next. And what's the future? I know nobody wants to see this, but it is going to be the future. It's going to be whole genome sequencing of babies. I know Mike Gelb talked yesterday, and there's a lot of pieces of that need to be addressed before it actually becomes reality, but we are starting to see some activity to actually do some research in that field. Next slide. We have two newborn screening by whole genome sequencing pilot projects that Travere is going to be supporting. One is with RTI International in North Carolina. They've been doing an early check program, screening for conditions not on the RUSP. They're now going to be doing whole genome sequencing. Tier 1 is going to include CTX. And tier 2 is going to require an additional consent, but they will put Zellweger's, maybe not all 14 of the PEX genes, but this is going to be a consented program looking at about 100 different conditions. They're looking at public health infrastructure and looking at public perception as far as acceptance of this new technology. And the other project is at Rady's Children's Hospital Institute for Genomic Medicine. Dr. Kingsmore, they just launched the Begin NGS for next-gen sequencing program, where they're going to be looking at about 388 different treatable conditions, including CTX and Zellweger's spectrum disorder. Travere is a sponsor, and they have this genome to treatment tool, which is going to have education and resources for providers and patients, which will talk about treatments. It will talk about patient advocacy. It will talk about the variants. They are working with Genomenon, which for Mike, I know their big thing is trying to flatten the VUS curve, which has to do with those variants of unknown significance. Every sponsor is going to have a seat at the table to continue to talk about the issues as they come around, because they will. The Rady's project is going to start in California and Tennessee, but they're going to try to expand throughout the United States. So these are just very exciting initiatives, in my opinion, to try to find these patients sooner rather than later. And I think that's it for me. Next slide. I think that's it. Thank you.